get this PowerPoint presentation up. Hopefully this isn't, well, I hope this isn't the last chapter that we go over, but it could be, depending on how far we get today. Okay. So, DNA. For a long time, I mean, subsequent to Mendel, right, we understood that there was information that was passed between parent and offspring, but we didn't really know what that molecule or what that information was. Mendel unequivocally showed that it's not the actual eyeballs that are being passed from mom to offspring or from dad to offspring, but rather it's information about those eyeballs, right? But really, there are two molecules that could potentially carry some information. One is DNA, and the other is protein. Remember, the way that DNA carries information is as a sequence of nucleotides, right? That sequence of A's, T's, G's, and C's carries some information. And when there is genetic variability between individuals, it's really the variability in that sequence of nucleotides. But proteins have variation as well, right? Because each protein consists of a sequence of amino acids. And there can be variability in that sequence of amino acids between individuals. And so subsequent to Mendel, we wanted to figure out which molecule it was that was actually carrying carrying information. So what we're going to do now is talk about a few experiments that show that DNA is this mechanism of inheritance. Now, I mean, to us who know that DNA carries this information, this isn't terribly groundbreaking, nor is it exciting. But at the time, it was a major avenue of research. So the first experiment that I want to talk about is the Griffith experiment. And in the Griffith experiment, researchers were working on Streptococcus, so the strep bacterium. And you guys know that strep is bad, right? You don't want to have strep. It makes you really sick. But there are variants of strep. There's a type S and a type R variant. The type S variant is virulent, particularly in mice. If you inject mice with the type S Streptococcus bacterium, it's generally lethal. But there is a second type, type R, that is non-lethal. And what makes one lethal and the other not lethal is the presence of, or absence of, some polysaccharide capsules. So remember way back to the beginning of the semester when we were talking about prokaryotic and eukaryotic cells. Prokaryotic cells, some of them had a capsule outside the cell wall and outside the plasma membrane. That capsule is a construct of polysaccharides, just really, really long, complex sugars. Now the S-type has one, and that makes it virulent. It makes it lethal. The R lacks one, makes it non-lethal. So what the Griffith research group did was they looked at different combinations of living and dead Streptococcus strains. So first of all, what they did was they took some S-type Streptococcus and they completely killed it. So they made sure that it was biologically inactive. And I have to mention that really you can't kill a bacterium because bacteria aren't considered living. They're these weird little entities that are just like packages of DNA that are able to inject their DNA into our cells and then usurp control over our cells so that we're replicating their genomes. Anyways, they deactivated these S-type bacteria and they injected it into mice, and they found that there was no virulence. There was no mortality in mice that were injected with S-type bacteria that had been deactivated. Then what they did, though, was they took some S-type bacteria that had been deactivated and they put it in a slurry with R-type bacteria, those non-virulent type, and they injected them together. So living R-type, dead S-type, and they injected them both into some mice. And what they found was that there was virulence, that there was some mortality in mice, which is interesting, right? Because the R-type bacterium shouldn't kill bacteria or shouldn't kill mice, and dead S-type bacteria also shouldn't kill mice. That's what that first experiment showed. But in combination, they did. So the question is, what happened? And the answer is something called transformation. Transformation is this phenomenon within bacteria where one can assume characteristics of the other. And they assume characteristics of the other by assuming some information from that other bacteria. Now what we know today is, what they're really assuming is DNA. So these non-virulent R-type bacteria were able to take the DNA of the S-type bacteria, even though they were dead, and they were able to replicate those gene sequences and create these polysaccharide capsules that made them virulent.
So the R-type took stuff from the S-type, which made it neutral?
Correct.
Okay.
Yep, that's exactly what happened. R-type took stuff from the S-type, which is interesting, I think. So what this showed was, between bacterial strains, you can have some exchange of information, but it didn't really solely identify that DNA as that mechanism of information exchange, right? That came with the next experiment, which is called the Avery experiments. And so what these guys did was they isolated molecule types. So in a slurry of bacteria, they pulled out all of the protein, and they had a highly concentrated and very pure solution of DNA. In fact, it was 99.98% pure. Very little stuff left over, just DNA. And they injected that DNA concentrate from the S-type bacterium into a slurry of R-type bacteria. And what they found was, again, transformation. But they didn't stop there. They chemically tested this pure concentration stuff that they extracted. And what they found was the stuff that was left over had the same chemistry as DNA. What that means is the stuff that they were actually injecting into the slurry of the living R-type bacteria was DNA, right? Secondly, it behaved very much like DNA did. So when they spun it in a centrifuge, it pulled out a solution at the same RPMs as DNA would. And when they ran it through a gel electrophoresis, it migrated very much like DNA did. Thirdly, when they exposed it to lipid and protein extraction protocols, nothing was pulled out. So when they spun it as fast as you would normally spin a centrifuge to pull proteins out, nothing came out. Same thing with lipids. Fourth, when they exposed them to enzymes that target and destroy things like proteins and RNAs, this stuff that was left over wasn't destroyed. So it couldn't have been a protein, and it couldn't have been an RNA. But when they did expose it to restriction enzymes specific for DNA, it was destroyed, it was digested. So the Avery experiments showed that this stuff that was actually transforming R-type bacteria into S-type bacteria was DNA. But really, the nail in the coffin came with this third and final experiment that I will tell you about. Let me, before I change slides, let me tell you a little bit about how it is that viruses work. So a virus, it looks like a lunar lander. So generally, they have some geometrical capsule that looks something like this. And it's three-dimensional. And it's attached to a long tube, and this long tube has legs, like this, some number of legs. What they do is they find some target cell, and then through this tube, and this protein capsule has their DNA, like this, just kind of floating around inside that capsule. And what they're able to do is they're able to find a target cell, insert this tube through the plasma membrane of the target cell, and inject DNA. But this capsule consists of protein. And so when people were thinking about viruses, specifically bacteriophages, which are viruses for bacteria, they thought, well, I mean, there could be information about this protein capsule that's being inserted. There could be protein that's being inserted from that capsule, or it could be the DNA. So the final experiment was called the Hershey-Chase experiments. And within the Hershey-Chase experiments, what they did was they tagged both proteins and DNA with different radioactive isotopes. So they tagged the DNA with radioactive phosphorus, and they tagged the proteins with radioactive phosphorus, and they tagged the DNA with radioactive sulfur. And then they exposed these bacteriophages to their target bacteria, and they saw what actually got transferred from the from the virus into the bacterium. And if I'd been referring to strep as a bacterium, that's correct, because it is. I almost confused myself there. Okay, so they were looking at viruses and seeing what viruses were actually putting in their target bacteria. And what they found was DNA was actually being transferred from that bacterial lunar lander into the bacterium. Okay, so that was a really long and drawn out way to say it's DNA that's actually transferring information from one individual to another. But even at that point, we didn't really understand what the nature of DNA was. And so there was this huge research effort to figure out what DNA actually consisted of, and then what it looked like, what the shape was. Okay, now most of this is going to be review, so please bear with me. But let's remember what DNA actually looks like. So DNA consists really of three things, right? There are those A's, T's, G's, and C's, which are called nitrogenous bases. There is a sugar, which is, in the case of DNA, deoxyribose, and then there's a phosphate. And those three things constitute a nucleotide. So that looks something like this, right? We have some sugar, which is ribose. I'm sorry, it's deoxyribose for the DNA, that's ribose for RNA. So it's deoxyribose. And then deoxyribose is attached to a large phosphate, right? And phosphate carries a charge, and that charge is negative, yeah. It's negative. And then coming off one of the carbons from our deoxyribose, there is a nitrogenous base. And those nitrogenous bases exist in two forms. They're either purines or they're pyrimidines. Purines are A and G, and then the pyrimidines are T's and C's. And the purines look a little bit different than the pyrimidines do, because there's a 5-member ring attached to a 6-member ring. And there are a bunch of double bonds that I won't need you guys to know about.
That you will need us to know about?
What's that?
That you will?
No.
Oh, okay.
They're called nitrogenous bases because at specific points they have nitrogens. And you guys, this is just like make-believe. This isn't really what an A looks like. But the point I'm trying to make is, within these rings there are nitrogen compounds, which is why they're called nitrogenous bases. And if there's a 5-member ring attached to a 6-member ring, that's either going to be A or G. Yeah, and that's a purine, so that would be an A. So this fellow named Chargaff looked at A's and T's and G's and C's, and he compared the relative numbers of A's, T's, G's, and C's, and he came up with what was called the Chargaff rule. And Chargaff's rule said there are going to be an equal number of A's and T's and an equal number of G's and C's. And what that suggests is when DNA is binding, when we have this formation of complementary strands, and please remember that the strands are complementary, they're not mirror images of each other. When we have these complementary strands of DNA, A is always going to bind to T, and G is always going to bind to C. Now, the relative proportion of A's, T's, G's, and C's, if it was a random assemblage, you would expect some equity in the number of A's, T's, G's, and C's, but there isn't. Within some species, there are going to be relatively more G's and C's. And one of the reasons for that is the bonds between G's and C's are much more stable than the bonds between A's and T's. Please remember that complementary strands are held together by hydrogen bonds. And hydrogen bonds are just a product of partial charge differentials, right? But the bonding between T's and G's actually form 3 hydrogen bonds, and the bonds between A's and T's only form 2 hydrogen bonds. And so in areas where there is a lot of thermal activity, like thermal hot springs, those organisms that are exposed to much higher temperatures will have a much higher concentration of G's and C's than they will of A's and T's. And that's because of the relative stability of those bonds.
So there's always going to be an equal number of A's, T's, and G's and C's. It's false.
They're correct. That is a false statement.
Okay.
But there will always be the same number of A's and T's, and there will always be the same number of G's and C's.
Okay.
So
Okay.
it's false totally, but separately there, it's true, because you have to have that complementary strand.
It's false that there will always be the same number of A's and G's. It is true that there will always be the same number of A's and T's. Okay. We okay with this idea so far? Okay, so enter Watson and Crick and Rosalind Franklin. Back in 1953, there was like this huge push to figure out the shape of the DNA molecule. They knew what the constituents were, but they didn't know how they arranged themselves in space, what the three-dimensional conformation of this was. Watson and Crick were on different sides of the Atlantic Ocean. James Watson was working in the US, Francis Crick was working in the UK. But they joined forces to figure out what the shape of the DNA molecule was. And they got a hold of this woman's research. His name was Rosalind Franklin. And what Rosalind Franklin did was she took what's called an X-ray crystallograph of the DNA molecule. And X-ray crystallography isolates some molecule, and then it blasts X-rays at it, and it looks at diffraction patterns. So how it is that these X-rays, which are very high energy, low